Morning from Brisbane. My name is Loan Nguyen from the University of Queensland. Today I would like to share with you our current publications in Frontiers in Genetics. Its title is An Epigenetic Aging Clock for Cattle using Portable Sequencing Technology. We concern about animal age because it will be useful for deriving important traits such as age at puberty. It also needs for breed registrations and making management decisions. However, it is very challenging to obtain in many farming conditions. There are several epigenetic aging clocks have been developed in humans, dogs, goats, and mice using arrays assay. We hypothesize that nanopore sequencing from unmodified DNA can also be used to co-methylation sites and predict age in cattle sample from very easy obtained sample like head. So in the study, we use group of 66 animals come from 3 different herds. Gene transfusion kit used for DNA extraction. Ligation kit was used for library prep and sequence on a mini device. Methylation site cost using F5C software. We apply 2 filtering options. The first to filter site that costs in at least 80% of animal, and the second option is to filter site close to genes report to humans and dogs from Wang et al. Study 2020. We also test for the gene associated from humans, dog, and cattle using linear mixed models. Methylation block was used to predict age, and 5-fold cross-validation was applied for age prediction accuracy. As you can see from figure 2, it's showing the sequencing coverage and total number of methylated site code for 66 samples. The number of sites was varies ranging from 2.8 million to 17 million sites. 3-fold coverage up to 10 million methylated sites were detected. The number of observed methylation sites also increased sharply with the sequencing coverage and then plateau at about 6-fold coverage. At figure 2, we're showing the significant age associated in humans, dog, and cattle. Out of 320 gene tests, there are 43 genes that are associated with age in cattle, suggesting they are important or potential candidates for the age prediction across species. Finally, blood was used to predict age of animals from the whole genome methylation data, and from the methylation data only near genes associated with age in human and dogs. The correlation of predict age and actual age in validation dataset was 0.71 and 0.6 for the dog human age associated size. The difference between the actual age and predict age for animal age less than 3 years are 1.4 years and was 1.5 years for animal age from 3 to 10 years. So in conclusion, methylation pattern change at cattle age and nanopore sequencing is a useful tool for profiling methylation. The 5 most significant age-associated genes could serve as promising candidates for age prediction clocks across species. This study achieved a prediction accuracy of 0.71, and the accuracy is expected to increase with larger number of sites. Finally, thank you so much for your listening, and please let me know if you have any comments or questions about our study.